In this last video for this week, I wanted to talk about how we grow animal viruses. We were talking about how we grow bacteria, especially in lab. We were saying we can grow them in broth cultures, we can grow them in on agar plates, or at least some bacteria. But viruses, remember, need a host cell. Need more than just food. They need cells. Why would we want to grow viruses? Well, you might need to grow a virus to identify them for clinical purposes, to help diagnose diseases. We might need to make viruses for vaccines, and actually, this is one of our biggest reasons why we grow viruses. And finally, there's all sorts of researchers that do research on viruses, so we might need to grow viruses for research purposes. There's three main ways that viruses are grown in the laboratory, and one of them is using live animals. This is a common way to grow viruses, but you can imagine that the complications and the restrictions of using live animals is significant. You need certain kinds of labs, you need humane procedures, and using live animals isn't always ideal. If you're studying a virus and how a disease progresses, it might be your only choice. But when we don't have to, we try to find another way to grow viruses. There's lots of, there's viruses for mice and rabbits and things like that. I actually worked in a research lab where we were studying a virus that infected caterpillars and was used for killing caterpillars for agricultural purposes. So sometimes we had a lot of caterpillars that we were growing up, and then we would inject them with the virus. So that's a live animal. I'm going to skip this one and go to this one next. Another common way to grow viruses in the laboratory is using cell cultures, or also called tissue cultures. So remember, viruses need host cells, and we can actually grow a lot of different host cells in the laboratory. And we isolate cells, we give them the right kind of media, and they can grow. So we can grow a lot of different kinds of human cells and cells from other organisms. What you can tell here is here's some animal cells growing, and they're normal cells. And once they're infected with the virus, it might take a few days, but the cells start looking kind of funny. And then you know that the cells have been infected and they're starting to die from that viral infection. Cell cultures are really useful. You can get cell cultures from all different organisms. And so if you wanted to grow cold viruses, you could get some cells from the upper respiratory tract and grow them in a cell culture. This is a really common way that's used for growing viruses and for research purposes, and when trying to identify unusual viruses. So cell cultures are common. It is relatively expensive. You need highly trained people to do it, but it's definitely a common way that we grow viruses in the laboratory. The third way that we grow viruses is using bird embryos, particularly fertilized chicken eggs. And the primary reason we use fertilized chicken eggs to grow viruses is to make vaccines. And the most common one made this way is the influenza vaccine. I just skipped to a picture I found on how vaccines are made in chicken eggs. And what they do is they have fertilized eggs, and then this looks like they put a little bit of iodine to sterilize them, and they inject some live active virus into the eggs that have been fertilized. As the chick embryo develops and the cells divide, the virus divides in them and eventually kills that developing chick embryo, but then the egg is kind of filled with, there's a lot of virus in there that will be isolated. And so influenza vaccines are made from these fertilized and chicken eggs. And it's a really common way to do it. It's relatively inexpensive and it works pretty well. You get really good vaccine out of it. One of the problems is if you have a sudden demand for a lot of vaccine, like we did with the COVID-19 pandemic, there's not just fertilized chicken eggs sitting around. You've got to get chickens to lay them. It works much better if you have kind of a routine thing going on. We know we're going to need a new influenza vaccine every year. So you're gearing up producing these eggs. And there's a lot of that goes into that to just get that whole process going every year. So just to reiterate, there's three major ways to make grow viruses in the laboratory. It's in live animals, in cell cultures, or in fertilized chicken eggs.